Hello and welcome back to the deep dive. Today we are not just reading a textbook. No, we are constructing a fortress.
A mental fortress.
A mental fortress, exactly.
Yeah.
One that's designed to withstand, the rigors of medical school exams and, of course, the high stakes of actual clinical practice.
Right.
So we're taking our stack of high-yield medical texts. Our main spine today is Murray's Medical Microbiology, and we're layering in some really crucial, nuanced details from Joette's, Melnick, and Edelberg's.
And the goal today, really, is to master a genus of bacteria that has, it's shaped history, it's terrified entire nations, and it has probably ruined countless dinner parties.
We are talking, of course, about the genus Bacillus.
It's just a fascinating genus because, a lot of the time in microbiology, we get bogged down in these long lists of organisms that all kind of seem to do the same thing.
Yeah, they blur together.
They absolutely blur together. But with Bacillus, we have this incredible, almost cinematic story of two siblings.
Two siblings. Okay, I like this. Are we talking like a Cain and Abel situation?
A little bit, yeah. A little bit. Imagine you've got a family portrait. On one side, you have the calculated, the highly dangerous, the infamous sibling that is Bacillus anthracis.
The bioterrorist.
Bioterrorist, the killer of livestock. It carries this absolutely terrifying reputation, the Black Bane.
And then standing right next to it in the portrait, looking almost identical under the microscope, is the opportunistic, maybe slightly sloppy scavenger sibling Bacillus cereus.
That's the one. Exactly.
Mm-hmm.
The one you are much more likely to encounter in your own kitchen, probably after leaving some rice out for a little too long.
So one kills you with a hemorrhagic mediastinitis, which I'm sure we'll get into that.
Oh, we will.
And the other just makes you regret your takeout order for about 24 hours.
That's the dichotomy. But the wild thing, and this is what's so interesting, is that genetically, they are remarkably similar.
Like, how similar are we talking?
Extremely similar. But understanding why they are so different clinically, despite that shared DNA, that is the absolute key to mastering this topic.
Okay, so that's our mission today. We're building a structure-function mental model. We need to be able to distinguish between these two using those high-yield, exam-tested buzzwords, things like motility, hemolysis, capsule type.
And we really have to get to the why behind the symptoms. Why does anthrax cause this massive life-threatening swelling?
Yeah.
And why does fried rice, of all things, make you vomit so quickly? We're going to dig right into those molecular mechanisms.
And it all starts with the shared family trait, right? The thing that defines the entire genus Bacillus and really separates it from so many others.
The survival bunker.
The endospore.
The endospore.
Let's start there. Murray puts this right at the top of chapter 20. The defining characteristic of Bacillus is that they are spore formers. Now, when I hear spore, my brain immediately goes to, like, fungi, mold, but this is different.
Very, very different. In bacteria, specifically in these two genera, Bacillus and Clostridium, the spore is not for reproduction. It is not about making more bacteria.
Okay.
It's all about survival. It's a state of suspended animation.
I like to think of it as a time capsule, or maybe even better, a doomsday bunker.
A doomsday bunker is the perfect analogy. So think about it this way. Bacillus species, they're gram-positive rods. They are everywhere. They're in the soil, in the water, in the air.
Ubiquitous.
Completely.
Right.
And in that state, they're vegetative. They're eating, they're dividing, they're metabolizing. But what happens when the nutrients run out? Say the soil dries up or the nitrogen levels just plummet. They don't just die.
They hit the panic button.
It's actually the opposite of panic. It's a highly calculated, highly ordered preservation sequence. It's called sporulation.
Okay.
The bacterium dehydrates itself. It strips itself down to the absolute bare essentials. We're talking just the DNA and a few critical enzymes. And then it wraps itself in multiple very tough layers of protein and membrane.
And Murray describes this resilience so vividly. These spores can withstand heat, radiation, desiccation, chemical disinfectants. You can boil them, and they survive. You can spray them with alcohol, and they don't care.
And Giavitz adds this detail that I always find a little bit chilling.
Yeah.
These spores can persist in dry earth for years, and not just a few years. We're talking decades.
Decades, seriously.
Oh, yeah. There are documented cases of spores being viable after 50, 60 years sitting in the soil. Some researchers even claim longer, like centuries.
That's incredible.
You could theoretically dig up soil that was contaminated during a cattle drive in the 1800s, and if those spores are in there, they're still viable. They are just waiting.
It's basically biological immortality.
In a sense, yeah. We call it a cryptobiotic state. There's no measurable metabolic activity. They aren't alive in the way we think of it, doing things. But they're absolutely not dead.
But they can't stay in the bunker forever if they want to reproduce, right? They need some kind of trigger to wake up.
Exactly. They need a signal that the coast is clear. And usually, that signal is the presence of water and specific amino acids,
Oh.
basically food.
Or in a clinical context, I'm guessing, a human host.
The warm, nutrient-rich environment of a human host.
Yeah.
That is the ultimate all-clear signal.
Okay, so before we dive too deep into the specific species, we have to make a really important taxonomic distinction right here. We're talking about gram-positive spore-forming rods. But there's another major genus that fits that exact description. Clostridium.
Yes, and this is a critical day one differentiator for any student. If you see gram-positive spore-forming rod on an exam, your brain should immediately see a fork in the road with two choices, Bacillus or Clostridium. How do you tell them apart instantly?
Oxygen.
Oxygen. It's that simple. Bacillus species are aerobes or facultative anaerobes. They like oxygen. They thrive in it. Clostridium species, on the other hand, are strict anaerobes. Oxygen is poison to them.
So if you've got a patient with a wound infection and you culture the bacteria and it grows happily out in the open air of the lab...
It's Bacillus. If it only grows in a special oxygen-free chamber, it's Clostridium.
Yeah.
That is your fundamental split.
Okay, got it. We have our bunker concept, we have our oxygen requirement. Now let's go back to the family portrait and look at the bad sibling, the calculated killer, Bacillus anthracis.
Bacillus anthracis, a true beast of an organism.
Let's visualize it first. If we were looking under a microscope, what are we seeing? Maria paints a pretty specific picture.
You are seeing very large gram-positive rods. And unlike some bacteria that clump up in clusters like grapes, that's staph or parap like Diplocacci, Bacillus anthracis has a very, very specific arrangement. Murray describes them as long serpentine chains.
Serpentine, like a snake. And Jawetz gives us that classic description, boxcars.
Yes, boxcars. They have these distinctive square ends. Most bacteria have rounded ends, sort of like a capsule pill. Anthracis ends are flat, almost like they've been cut off. So when they line up end to end, they look exactly like a train of boxcars.
That's a great visual anchor to hold on to. Square ends, long chains. Now, here's where it gets really interesting, and this is a point that trips people up on exams constantly. We just spent 5 minutes talking about how important spores are to this genus. But if I take a blood sample from a patient with active anthrax, or maybe a fresh tissue biopsy and I gram stain it, do I see any spores?
No. Yeah. You will never see spores in a fresh clinical specimen.
Never, even though it's a spore former.
Never. And you have to understand the why to remember this. Go back to the bunker analogy. You only go into the bunker when things are bad outside. You don't live in the bunker when you're staying at a 5-star resort.
And the human body is the 5-star resort.
Exactly. Inside a host, it's warm, 37 degrees Celsius. It's wet. There are plenty of nutrients. The bacteria are in their vegetative state. They're growing, they're dividing, they're causing all sorts of havoc. They have absolutely no biological reason to waste energy making a spore.
So if I see a slide that's labeled anthrax and it has spores on it, what does that tell me about where that sample came from?
It tells you that the sample is old. It's from a culture. If that culture plate sits on the lab bench for 2 or 3 days, the nutrients in the agar start to run out. The bacteria sense that starvation, and then they sporulate. So spores are an artifact of culture, not a finding in fresh tissue.
That is a crucial spore rule to remember. Okay, let's talk weapons. Bacillus anthracis isn't dangerous just because it's a rod. It's dangerous because it's carrying some heavy artillery.
We call it the double-barreled weapon system. The virulence of anthrax, all of it, depends entirely on 2 specific plasmids.
And just a quick refresher, plasmids are those little extra circles of DNA that bacteria can kind of swap around, right? Like downloadable content for a video game.
That's a perfect way to put it. They aren't part of the main chromosome. They are extra genetic packages. And for B. anthracis, it has 2 of them that matter, PX01 and PX02.
Okay.
And here's the kicker. If you cure the bacteria of these plasmids, if you can somehow remove them, the bacteria becomes completely harmless. It essentially just becomes a B. cereus without the food poisoning toxins.
So the lethality is entirely contained in these 2 downloads. Let's break them down.
Yeah.
Weapon number 1, the shield. This is encoded on plasmid PX02.
PX02 encodes the capsule. Now listen very closely here because this is an extremely high-yield differentiator. If I asked you what the capsule of, say, strep pneumo or meningococcus is made of, what would you say?
Polysaccharides. Sugars. Almost all bacterial capsules are made of sugars.
Right. That's the standard bacterial armor. But Bacillus anthracis is different. It has a protein capsule, specifically a polypeptide capsule.
And what's it made of?
It's composed of poly-D-glutamic acid.
Poly-D-glutamic acid. Okay, why does the D matter?
This is the elegant evil of biochemistry. Most amino acids in nature, the ones in your body, in your enzymes, they're L-isomers, L-glutamate, L-alanine. Our immune system's enzymes, our proteases, are designed to recognize and cut L-amino acids.
Right.
They're like scissors that are designed only for right-handed paper.
And anthrax is using left-handed paper.
Exactly. By building a capsule out of D-glutamic acid, anthrax creates kind of invisibility cloak. Our proteases physically cannot lock onto the shape of those D-amino acids to digest them, so the capsule stays intact, and it masks the bacteria from phagocytosis. The macrophages just slide right off it.
That is incredibly clever. It's using chirality, molecular handedness as a defense mechanism.
It is. So PXO2 is the shield of the poly-D-glutamate capsule.
Okay, so that's the shield. Now let's talk about PXO1, the sword.
PXO1 codes for the toxins, and this is a fascinating biological system. It's what we call a tripartite toxin, meaning it has 3 parts. But here's the catch. None of the 3 parts are toxic on their own.
They have to assemble, like Voltron, or what's a better analogy?
I like to think of it as a Lego model.
Yeah.
You have these 3 individual proteins just floating around: protective antigen, PA, edema factor, EF, and lethal factor, LF. Individually, they're harmless, but together they're deadly.
Okay, let's walk through the mechanism step by step. Let's call this the Lego model of toxicity. Step 1 has to be entry. That's the job of the protective antigen, right?
Correct. Think of protective antigen, or PA, as the key.
Yeah.
Or maybe the Trojan horse. PA floats through the blood, and it binds to specific receptors on host cells. And the scary part is, these receptors are everywhere: brain, heart, intestine, lung, muscle.
So the PA binds to the cell surface. What happens then?
Then a host enzyme called furin comes along and cleaves the PA. It just snips off a small piece, leaving a fragment called PA63 stuck to the cell surface.
So our own body helps arm the weapon.
It is. It's practically inviting the vampire in.
Yeah.
Once these PA63 fragments are cleaved, they can slide across the cell membrane and find each other. 7 of them come together to form a ring, a heptamer.
A heptamer ring. That sounds like a portal or something.
It essentially is a portal. It's what we call a prepore. This ring acts as a docking station. It just sits there open and waiting. And what's it waiting for?
The other 2 Lego pieces: edema factor and lethal factor.
Right. EF and LF come along and bind to this ring. Then the cell, thinking it's bringing in something useful, performs endocytosis on the whole complex. It just swallows it into a little bubble called an endosome.
So now they're inside the cell, but they're trapped in this bubble.
Correct. But here's the final trick. The cell naturally acidifies the endosome. It lowers the pH inside. This drop in pH triggers the PA ring to undergo a conformational change. It snaps open and punches a hole right through the endosome membrane, injecting EF and LF directly into the cytoplasm.
That is a heist movie level of entry. The protective antigen builds the door, the cell swallows the door, and then the door opens from the inside to release the toxins.
Precisely. And now, once they are in the cytoplasm, the real damage begins. Let's talk about the first combination: PA plus edema factor.
This creates edema toxin. Edema factor, EF, is an enzyme. Murray calls it a calmodulin-dependent adenylate cyclase.
That's a mouthful, I know, but let's unpack it because it's super high yield. Adenylate cyclase is an enzyme that makes a signaling molecule called CAMP cyclic AMP.
And we've seen this mechanism before, haven't we? This should sound familiar.
We absolutely have. It's the same basic strategy used by Bordetella pertussis and Vibrio cholerae. So the EF from anthrax binds to a host protein called calmodulin to activate itself, and then it just goes crazy, converting all the cell's ATP into KMP.
And when intracellular CMP levels skyrocket like that, what happens to the cell?
The cell essentially gets confused and panics. In simplified terms, high KMP is a signal for the cell to dump ions, and with them, water. It just completely disrupts cellular homeostasis, so fluid rushes out of the cells and into the extracellular space.
Which leads to edema.
Massive, massive edema. Swelling that can be so severe it compromises blood flow or even crushes organs.
So EF is the pump. It pumps fluid out of the cells. Now, what about the other combination? PA plus lethal factor.
This creates lethal toxin, and this one is the blade. Lethal factor, or LF, is a zinc-dependent protease.
A protease cuts proteins, so what is its target? What is it cutting?
It cuts a protein family called MAP kinase, mitogen-activated protein kinase.
MAP kinase. That's a huge part of cell signaling, isn't it? It's like the cell's main switchboard.
Exactly. The MAP kinase pathway tells the cell to grow, to divide, and fundamentally to survive. It controls the entire stress response.
Mm.
Lethal factor basically walks into the control room of the cell and just cuts the main cables.
So the cell just goes silent?
It goes silent and then it dies. It triggers apoptosis and necrosis. But it also does something else that's really sinister. By disrupting this pathway, particularly in macrophages, it triggers a massive, unregulated release of inflammatory cytokines, things like TNF-alpha and IL-1 beta.
The cytokine storm.
The cytokine storm. So you get this combination of widespread cell death combined with massive systemic inflammation. This is what ultimately leads to shock and tissue destruction.
Okay, I want to recap this toxin mechanism because this feels like the absolute core of any exam question. PA is the key that builds the doorway.
Right.
EF is the pump that causes swelling by cranking up HAM-P.
Yep.
And LF is the blade that kills the cell by cutting MAP kinase.
Perfect. If you can visualize that, the key, the pump, and the blade, you will answer every single anthrax toxin question correctly.
Okay, so we know what the organism looks like, the boxcars, and we know how it kills the Lego model. Now let's talk about how it gets to us. Part 4, epidemiology.
The first thing you absolutely have to understand is that humans are accidental hosts. Bacillus anthracis doesn't want to be in humans. Its natural life cycle is in herbivores, cattle, sheep, goats.
Right, they eat the spores from the soil while they're grazing.
Exactly. And so, historically, this was an occupational disease for humans. We called it wool sorter's disease.
Because people processing goat hair or animal hides would inhale the spores from the contaminated material.
Precisely. But in the modern era, when we say anthrax, most people don't think of sheep shearing anymore. They think of bioterrorism.
And for very good reason. I mean, the spore is the perfect biological weapon. It's incredibly stable, it can be aerosolized, and it is brutally effective. And Murray highlights 2 major historical data points here. First, Sverdlovsk in 1979.
This is a haunting story. Sverdlovsk was a city in the Soviet Union. In April of 1979, people started dying mysteriously of this rapidly fatal pneumonia. The official Soviet government line was that it was bad meat intestinal anthrax from contaminated food.
But that was a lie.
It was a complete cover-up.
Yeah.
It wasn't until years later, after the fall of the Soviet Union, that the truth came out. There was a secret military facility, Compound 19, that was producing anthrax spores for weapons.
Wow.
And a worker removed a clogged filter on an exhaust vent and just forgot to replace it. A simple mistake.
Just a simple mistake.
The next shift came in and turned the machines on. For several hours, anthrax spores were pumped into the night air. The wind that night was blowing southeast. It blew right over a ceramics factory and other industrial plants.
And the workers there.
They breathed it in on their way to work. Downwind from that facility, at least 79 people contracted inhalation anthrax. 68 of them died. It was a terrifying real-world experiment that proved exactly how lethal the aerosolized form is.
And then, much closer to home, the 2001 U.S. Postal Service attacks.
This changed everything for the U.S. medical and public health system. Letters containing highly refined, weapons-grade anthrax spores were mailed to news media outlets and 2 U.S. senators.
I remember that so clearly, the fear of just opening your mail.
It showed that you didn't need a missile or a bomb, you just needed an envelope. The spores were so fine they behaved almost like a gas. They just leaked out of the envelopes during the automated mail sorting process. In the end, we had 11 inhalation cases and 11 cutaneous cases. 5 of the 11 inhalation patients died.
And this really brings us to the 3 routes of infection. The way the spores enter your body determines the type of disease you get.
Correct. The 3 routes are inoculation, which causes cutaneous anthrax, ingestion, which causes GI anthrax, and inhalation.
Let's walk through the clinical presentation of each of those, starting with the most common one in nature, cutaneous anthrax.
This accounts for about 95% of all natural cases. You're thinking of a farmer, a veterinarian, someone handling animal hides. The spores get into a small cut or an abrasion.
It produces a very, very specific lesion, the coal lesion.
Right. The name anthrax actually comes from the Greek word for coal, anthracis. The lesion starts as a small papule. It looks kind of like a bug bite. Then it develops into a vesicle, a little blister, and finally that ulcerates and turns into a necrotic lesion with a depressed black center, a black eschar.
Okay, now here is the million-dollar clinical question that you'll see on every exam. Does it hurt?
No. That is the key differentiator. The anthrax eschar is painless.
That is just so counterintuitive. You have this black, necrotic, rotting hole in your arm, and you're saying it doesn't hurt.
It might be itchy, but it is characteristically not painful. The toxins, especially lethal toxin, actually destroy the local nerve endings. So if a patient comes into the ER with a nasty-looking boil and they're screaming in pain, you should be thinking staph or strep. If it's numb and black, you have to think anthrax.
And there's usually massive edema all around it, right? Thanks to our friend edema toxin.
Right. The swelling looks completely disproportionate. You'll see a small black spot, but the entire arm or limb is massively swollen. If it's left untreated, the mortality is about 20%. But with antibiotics, it's very rarely fatal.
Okay, let's move to the second route, gastrointestinal anthrax.
This one is very rare in humans, but it's common in animals. It happens when you eat undercooked meat from an infected animal.
It sounds horrifying.
It is. The bacteria invade the mucosa of the GI tract. You can get ulcers in the mouth or the esophagus, which leads to this massive neck swelling from regional lymphadenopathy. Or the organism can invade the terminal ileum and the cecum in the lower gut.
And the symptoms?
Nausea, vomiting, bloody diarrhea. It progresses incredibly rapidly to systemic disease, to sepsis. The mortality rate here is close to 100%, simply because it's so aggressive and it's so hard to diagnose in time.
And finally, the third route, the bioterror route, inhalation anthrax, also known as the silent killer.
This is by far the most dangerous form. And the really scary part is its two-phase progression.
Okay, walk us through phase one. You breathe in the spores, they go deep into your lungs.
Right. The spores are tiny, about 1 to 5 microns, so they bypass all the normal defenses, the hairs and the mucus, and they go straight down into the alveoli. The alveolar macrophages, the little vacuum cleaners of the lung, they pick them up. Now, listen carefully to this part. The spores do not germinate in the lung tissue initially. They don't cause a pneumonia right away.
That's a huge distinction, so it's not a pneumonia at first.
Not at first. The macrophages act like a taxi service. They pick up the spores, and they carry them to the mediastinal lymph nodes, the lymph nodes in the center of your chest. That is where the spores germinate. That is where they start making toxins.
So during this travel time, during phase one, the person just feels kind of like they have the flu.
Exactly. Fever, muscle aches, malaise, a dry cough. It's completely nonspecific. You just think you have a viral syndrome. This is the latency period, and it can last for days or even, in some cases, weeks.
And then comes phase two, the fulminant disease.
The toxins reach a critical mass in the lymph nodes. The nodes begin to hemorrhage and necrose. The bacteria then break out of the nodes and spill into the blood. This is what leads to the classic radiographic sign of inhalation anthrax.
The widened mediastinum.
Yes. On a chest x-ray, the space between the lungs, the mediastinum, looks abnormally wide. And it's not just swollen with fluid, it's filled with blood. It is a hemorrhagic mediastinitis.
So if you see widened mediastinum on a case study or an exam question in the context of a patient with fever and sepsis, you have to mark anthrax.
It is pathognomonic in that context. The patient develops high fever, massive edema, septic shock, and often meningitis. About 50% of inhalation cases end up spreading to the meninges.
And once phase two starts, the clock is ticking really, really fast.
It's more than ticking. It's practically run out. Shock and death usually occur within 3 days of the onset of phase two. The toxins have already done the damage. Even if you give antibiotics and kill all the bacteria, the toxins that are already circulating continue to do their work.
That is just terrifying. Okay, so we suspect anthrax. We need to diagnose it. Part 6, the lab bench.
First rule of the anthrax lab. You have to alert the lab. If you suspect anthrax, you do not just send a sample down the pneumatic tube. You do not want a lab tech opening and sniffing a plate of Bacillus anthracis on an open bench. It creates an aerosol risk. It has to be handled in a biosafety cabinet.
Right. Safety first. Once it's safely in the cabinet, what are we looking for on the culture plate? We need to be able to tell it apart from its, less harmful cousins like Bacillus cereus.
There are 3 gold standard characteristics for B. anthracis culture, and Murray really hammers these home. Number 1 is hemolysis.
Or the lack of hemolysis?
Correct. Bacillus anthracis is non-hemolytic on sheep blood agar. This is a crucial distinction. B. cereus and most other environmental Bacillus species are beta-hemolytic. They produce a clear zone around their colonies because they destroy the red blood cells. Anthracis leaves the red cells alone.
Okay, so anthrax means no hemolysis. Cereus means hemolysis. What's number 2?
Motility. Bacillus anthracis is non-motile. I like to think of it as a lazy killer. It doesn't need to swim around. It lets the host's body carry it where it needs to go. If you look at it in a test tube of semi-solid agar, it only grows straight down the stab line. It doesn't swarm out into the media.
And you contrast that with B. cereus.
B. cereus is motile. It swarms all over the place. It's very active.
And number 3 is the actual colony appearance itself.
Right. It has a very characteristic ground glass surface texture,
Mm-hmm.
and it's incredibly sticky or tenacious. If you touch a colony with a sterile loop, it stands up in a little peak. It doesn't smear. It looks like beaten egg whites.
And Jawwitz adds a great visual for this too, the medusa head.
Yes, if you look at the colony under low magnification, the edges have these little curled projections that look like locks of hair or maybe snakes. So medusa head or curled hair appearance.
Okay, let's summarize the lab diagnosis for anthrax because this is pure exam gold. Non-hemolytic, non-modal, sticky ground glass colonies with a medusa head.
And if you need 100% confirmation, you can use the gamma phage test. This is a virus, a bacteriophage, that specifically infects and lyses B. anthracis. So if the phage kills the bacteria, it's anthrax. Or you can use a DFA, a direct fluorescent antibody test, to look for that unique poly-D-glutamic acid capsule.
Okay, we've diagnosed it and now we have to treat it. Part 7, treatment and prevention.
For treatment, early intervention is absolutely everything. The antibiotics of choice, especially in a bioterrorism scenario, are ciprofloxacin or doxycycline.
Wait, hold on. Isn't penicillin effective? I mean, it's a gram-positive rod. Penicillin should work, right?
Historically, yes. Naturally acquired anthrax, from sheep or goats, is often sensitive to penicillin, and you can use amoxicillin. But, and this is a very big but, if you are dealing with the potential bioterror strain, you have to assume it has been engineered.
Engineered to be resistant to our first-line drugs.
Exactly. The Soviets and other state programs specifically worked on creating penicillin-resistant strains. So the guideline now is, do not start with penicillin. You start with Cipro or Doxy.
And for something as serious as inhalation anthrax, you don't just use one drug.
No, you use a cocktail. You use combination therapy. You add a second or even a third agent like rifampin, vancomycin, or clindamycin. You need to hit it as hard as you can to stop the bacterial replication as fast as possible.
And clindamycin has an added benefit there, right? It's a protein synthesis inhibitor.
Yes, that's a key point. By stopping protein synthesis, clindamycin can help shut down toxin production. It helps turn off the faucet, so to speak.
But what about the toxins that are already out there? I mean, if the bacteria are dead, but the toxins are already floating around, antibiotics won't help with that problem.
And that's where the modern therapies come in. We now have monoclonal antibodies, specifically one called raxibacumab.
How does that work?
It's an antibody that is engineered to bind directly and specifically to the protective antigen, to PA. If you gum up the PA, it can't bind to the cell receptor, and it can't form the pore. You essentially neutralize the entire delivery system.
That's amazing. It's a true antidote. Okay, what about prevention? Is there a vaccine?
There is. It's called Biothrax. It's what's known as an adsorbed vaccine made from the cell-free filtrate of a culture. And what it essentially contains is the protective antigen protein.
So it trains your body to recognize the key.
Exactly. If you have antibodies against PA, you are immune to the toxin's effects. But it's not a vaccine for the general public. It requires a long series of injections and regular boosters. It's really reserved for the military and people in high-risk occupations, like researchers or wool workers.
Okay, that covers the capulated bioterrorist. Now let's pivot to the other sibling. The one who didn't go to military school but still causes a lot of trouble. Bacillus cereus.
Bacillus cereus, the party animal of the family, the opportunist.
So let's look at its profile. How does it differ from anthrax in the lab? We already dropped some hints earlier.
It is the opposite of anthrax in the lab. It's easy to remember. Anthrax is non-modal, cereus is modal, anthrax is non-hemolytic, cereus is beta-hemolytic. Anthrax has that special protein capsule. Cereus does not, for the most part.
And while anthrax hides in wool and animal hides, cereus hides in food, specifically rice.
This brings us to clinical syndrome number 1, gastroenteritis. We often call it fried rice syndrome. But we need to be very careful here because B. cereus actually causes 2 distinct types of food poisoning. And Murray makes a big deal about this distinction because the mechanisms are totally different.
Okay, let's break them down. Form A, the emetic form. That means vomiting.
Right. This is the one that is classically associated with fried rice. Here's the scenario. You cook a big pot of rice for a stir fry. The boiling water kills all the vegetative bacteria. But what survives...
The spores, the bunker.
The spores survive the boiling. Then, you leave the pot of rice out on the counter to cool down for a few hours before you put it in the fridge. This is the danger zone.
Because the rice is warm, it's wet, it's full of starch.
It is a bacterial paradise. As the rice cools down into that perfect temperature range, the spores germinate. They turn back into active bacteria and they start feasting on the rice. And as they grow, they produce a specific toxin called cereulide.
Now, this is the crucial part. Is the patient eating the bacteria or are they eating the toxin?
In the emetic form, It is an intoxication.
Mm.
The bacteria have already made the toxin in the rice. You
Yeah.
eat the rice, you are eating the preformed toxin. And that is why the onset is so fast. We're talking 1 to 6 hours after eating. You don't have to wait for the bacteria to grow inside your gut.
And because it's a preformed toxin, reheating the rice doesn't help you.
Not at all. The emetic toxin, cereulide, is heat stable. You can throw that rice in a wok and fry it again in high heat, and the toxin will survive perfectly fine. It then acts on the vagus nerve to trigger vomiting.
So rapid onset vomiting caused by a heat-stable toxin found in rice. What about form B, the diarrheal form?
The diarrheal form is different. This is usually associated with things like meat dishes, vegetables, or sauces. In this case, it is an infection. You ingest the bacteria or the spores, they survive the acid in your stomach, they reach your small intestine, and then they start to grow and produce the toxin inside your gut.
And since the bacteria have to grow first, the incubation period is longer.
Yes. Usually more than 6 hours, sometimes up to 16. And the toxin here is different. It is a heat labile enterotoxin. It's destroyed by cooking.
And what's the mechanism?
It acts just like the edema factor of anthrax or the cholera toxin. It increases intracellular CMP.
Yeah.
This causes a massive flow of water out of the intestinal cells, leading to watery diarrhea.
Okay, let me try to recap this because it's important. Emetic form. Think rice, vomiting, fast onset. That's an intoxication with a heat-stable toxin.
Perfect.
Diarrheal form. Think meat or sauces, diarrhea, slower onset. That's an infection with a heat labile toxin that works via KP.
You've nailed it. Most of the time, these are self-limiting. You have a really bad day, you rehydrate, and you recover. But B cereus has a much darker side. It's not just about bad takeout.
Clinical syndrome number 2, ocular infections. This just sounds nasty.
It is devastating. This typically happens with penetrating trauma to the eye. Say a metal worker gets a splinter of metal in their eye, or a farm worker gets hit with a soil-contaminated object like a tree branch.
So the bacteria get injected directly into the vitreous of the eye.
And they go to work immediately. B cereus is everywhere in soil. Once it's inside the eye, it produces a trio of destructive toxins: an necrotic toxin, a hemolysin called cereolysin, and phospholipase C, which is a lecithinase.
It basically just melts the structures of the eye.
It's a fulminant panophthalmitis.
Yeah.
The destruction is incredibly rapid. We are talking about the potential for complete loss of the eye within 48 hours. If you see a post-traumatic eye infection that is just exploding with inflammation, you have to think B cereus, and you have to act incredibly fast.
And there's one more really weird syndrome that Murray mentions, the anthrax mimic.
This is a truly scary development in microbiology. There have been several documented cases, specifically in welders and other metal workers in Texas and Louisiana, of a severe pneumonia that looked for all the world exactly like inhalation anthrax.
But it was actually B cereus?
It was B cereus.
It's serious.
It was a B cereus that had acquired the PX01 plasmid from B anthracis.
So it had the anthrax toxin gene.
Yes. It was an encapsulated strain of B cereus that was producing the anthrax toxins. And it killed patients, just like anthrax does. It's a terrifying example of how these plasmids are mobile. The genetic line between the harmless sibling and the killer sibling is getting blurry.
Wow. That leads us very nicely into Part 9, treatment for B cereus. We know anthrax is treated with Cipro, Doxy, sometimes penicillin. What about cereus?
Here is a critical resistance alert for everyone listening. Bacillus cereus naturally produces beta-lactamases.
So penicillin is completely out.
Penicillin is useless. Cephalosporins are useless. Ampicillin is useless. If you try to treat that traumatic eye infection with a cephalosporin, that patient is going to lose their eye.
So what is the drug of choice?
You need to go to vancomycin or clindamycin. Sometimes you might add an aminoglycoside like gentamicin for synergy, but the core of your treatment has to be Vanoc or Clinda.
And that is a huge differentiator from anthrax, which is usually penicillin sensitive, at least in its natural form.
Correct. Let's state it clearly for the exam. Anthracis, penicillin sensitive, usually. Cereus, penicillin resistant, always.
Okay, I think we have built the fortress. We've analyzed all the bricks. Let's do a quick structural check before we move on to the quiz.
Let's summarize the big take-home points.
The genus. Bacillus means spore-forming and aerobic.
Anthracis. Think non-modal, non-hemolytic, and that unique protein capsule made of poly-D-glutamate.
Anthrax toxins, the triad. PA is the key. EF causes edema via KMP, and LF causes death by cleaving MAP kinase.
Cereus. Think motile and hemolytic. It causes food poisoning, the rice vomit stable toxin form versus the meat diarrheal ball toxin form.
Serious treatment. It's resistant to penicillins. You have to use vancomycin.
Perfect. And here is a provocative final thought for you to mull over. We mentioned those metal workers in Texas who died from B. cereus, those carrying anthrax toxins. We know these bacteria share environments. They live side by side in the same soil. We know plasmids can move between species. Now, B. cereus is everywhere. It's in your kitchens, it's on your skin, it's in the dirt. B. anthracis is actually quite rare. But if B. cereus, this ubiquitous scavenger, starts routinely picking up the PX01 plasmid from its dangerous cousin, are we looking at a future where fried rice syndrome could become a lethal hemorrhagic event? It really challenges our neat little boxes of pathogen versus opportunist.
That's a sobering thought. The microbial world is far more fluid and dynamic than our textbooks sometimes suggest.
On that happy note,
Mhm.
are you ready to test your fortress? It's time for quiz mode.
Yeah.
I am going to read a rapid-fire clinical scenario or a direct question. Pause for a moment, think of the answer, and then our expert will reveal the truth. Ready?
Let's do it.
Question 1. A 35-year-old textile worker presents with a painless black necrotic lesion on his forearm, surrounded by massive edema. A biopsy reveals large gram-positive rods and long chains. Which unique structural component is responsible for preventing phagocytosis of this organism?
The answer is a polypeptide capsule composed of poly-D-glutamic acid. Remember, most capsules are sugar. Anthrax uses protein, specifically D amino acids, which our host proteins cannot degrade.
Question 2. Regarding the toxins of Bacillus anthracis, which component acts as the binding moiety that allows entry of the other toxic components into the host cell?
The answer is protective antigen, PA. It binds to the receptor, it gets cleaved, and it forms the heptamer pore that allows the other factors to enter. It is the key.
Question 3. A group of students develops rapid onset vomiting about 3 hours after sharing reheated fried rice at a party. Which statement best describes the pathogenesis of their illness?
The answer is ingestion of a preformed heat-stable enterotoxin, which is cerulide. This is an intoxication, not an infection, which explains the very rapid onset.
Question 4. You are examining a blood culture from a patient with suspected inhalation anthrax. Which set of laboratory characteristics would confirm the identity as Bacillus anthracis over the more common Bacillus cereus?
The answer is non-modal and non-hemolytic. Remember, B. cereus is modal and beta hemolytic. Anthrax is the lazy non-hemolytic killer.
Question 5. What is the specific biochemical mechanism of action for the edema factor, EF toxin in anthrax?
The answer is that it is a comodulin-dependent adenylate cyclase that increases intracellular CAMP. This high level of CAMP leads to a massive efflux of fluid from the cell, causing edema.
Question 6. A patient with suspected inhalation anthrax has a chest X-ray that shows a prominently widened mediastinum. What is the underlying pathology that causes this classic radiographic sign?
The answer is hemorrhagic mediastinitis and massive enlargement of the mediastinal lymph nodes. It is not initially a pneumonia. The primary pathology is in the lymph nodes in the center of the chest.
Question 7. Which class of antibiotics is Bacillus cereus intrinsically resistant to, rendering them ineffective for treating a condition like traumatic endophthalmitis?
The answer is penicillins and cephalosporins. B. cereus produces broad-spectrum beta-lactamases. You must use something like vancomycin or clindamycin instead.
And our final question, question 8. The lethal factor, LF, of anthrax is a zinc-dependent protease that cleaves which specific intracellular target to cause cell death?
The answer is MAP kinase, or mitogen-activated protein kinase. Cleaving this critical signaling protein disrupts cell function, leading to apoptosis and a massive inflammatory response.
And that's a wrap on our Bacillus deep dive. You have now hopefully fortified your mind against the spores.
Keep reviewing those toxin mechanisms, the key, the pump, and the blade. They are absolute high-yield gold for your exams.
We'll see you on the next deep dive. Stay curious.